Very good morning, students. So in this lecture we are talking about the physiology of Sycon. So in physiology we have different systems which are very comparable to the previous animals. So first of all, if we talk about the movement, if we talk about the movement in Sycon, so the Sycon, it is not possessing any kind of locomotory organs. So there are no specialized organs for locomotion. So as it is sessile in nature, it is attached to the substratum by a base. So it is not moving here and there. So it is not showing any kind of locomotion as per the displacement is concerned. But with the water currents, the cylinder of the Sycon, it can move in different directions along with the water. So this is somewhat of the movement which is shown by the Sycon cylinder. Now, what about the movement of the body layers of the Sycon? So, the whole body, it may show slow contractile movements as the outermost layer or the pinacoderm is highly contractile, or it can contract to some extent. So, the movements of the cells, uh, they are amoeboid in character rather than muscular. So here what happens that the outermost layer of the body can contract and relax to some extent. So the whole cylinder of the Sycon can move uh, to some extent in the water. So that was all about the movement of Sycon in the environment. The next thing is the water circulation. So as we have discussed in the previous lectures also, this water circulation is maintained by the flagellar movements of the choanocytes. So all the flagellae of the choanocytes, they move inside the body of the Sycon, which are producing a water current which expels the water out from the osculum. So ultimately, when the water goes out of the osculum, there is an empty space which is formed inside the spongiocele. So suppose this is the Sycon, and in the very center you have the spongiocele. So water is entering into the body through ostia. So ultimately the water is reaching in the spongiocele in the very center. So when the choanocytic flagellae they move, the water is expelled out through the osculum. So this water circulation is maintained by the choanocytes. So what happens when the water moves out? When the water is moving out of the osculum, what happens? That makes an empty space inside. So this space has to be filled. So inside, if the Sycon is present in water, there cannot be an empty space. So what happens? The water will move inside the body of the Sycon. So the water will be replenished after some time. So this is the course of circulation. Next we have the nutrition part. So as we have discussed previously that the choanocytes, they are responsible for the intake of the food, and we have discussed that if this is a choanocyte and it has got a flagellum in the very center, so what is happening, this is the collar of the choanocyte, and this collar is made up of many microfilaments. So I'm just making 4, but these are quite large in number. They can be 16, they can be even more. So what happens is when the flagella, it shows a movement and the water is driven through the collar, and then the food is filtered, and this food ultimately it reaches the center over here inside the collar. Now this food is taken into a a food vacuole which is formed by the pinching of the membrane. So this food vacuole, it has now the food inside it. So now it can pinch off and it can come inside the body of the choanocyte, and this vacuole is containing the food. Now these are the food particles which are present, and further the digestion will occur with the help of the lysosomes. So that we have already discussed in the previous animals. So the process of the digestion is altogether the same, but here it is the partial digestion as we have discussed. So partially digested food, it will go to the mesenchyme. So ultimately it will go to the mesenchyme, and here an amoebocyte will receive this food, and this amoebocyte will be called as the thesiocyte. So that will be a food storing cell. So what we are concerned here is with the nutrition. So here the nutrition is due to the choanocyte, and the food taken is filtered. So that's why the Sycon is also called as a filter feeder. So that was about the nutrition. Now we are talking about the respiration process in case of Sycon. So this is a very similar kind of process which we have discussed in amoeba or paramecium. So here what happens, if this is the Sycon, and each of its cell will respire on its own. So what happens that each and every layer, or you can say each and every cell of the two layers, or the pinacoderm or the choanoderm, they all respire by their own. So what happens that you have dissolved oxygen outside. So here you have the dissolved oxygen. So I'm writing DO2. So it is high in concentration in the water. So it is going towards the cells. So if this is the outermost layer, suppose this is the pinacoderm, this is the outermost layer. So these cells will be taking this dissolved oxygen simply by diffusion. So diffusion is the process by which it is going inside the cells, and what will come out, it will be the carbon dioxide. So this is a very similar process which is going on in amoeba, it is going on in paramecium. So here we have to consider each and every cell is respiring on its own. So there is no specialized respiratory system as such in Sycon. So that was all about respiration. If we talk about the excretory process in case of Sycon, so again this is very similar to the previous animals. So the proteins, they are breaking down into amino acids which are giving you the nitrogenous waste product. So the removal of this nitrogenous waste product is called as the excretion. So what happens that when the ammonia, it is formed inside the cells of the Sycon, so this ammonia is converted into ammonium ions. So we have previously discussed this equation, that ammonia, it joins water and it is forming ammonium ions, NH4 plus, and it is forming OH negative. So this is the reaction. So this particular ammonium ion which is present now in the cells, it will be given out into the environment. So if this is a particular cell or a pinacocyte, so inside a pinacocyte the ammonium ions are formed, and those will be given out into the environment or into the water simply by diffusion. So again this is the same process that we have discussed in the previous animals or the protozoans. Here some scientists, they think that amoebocytes, amoebocytes which are present in the mesoglea, they are helping in excretion. So what they say is that amoebocytes which are present in the mesoglea, they will retain this particular excretory product inside them for some time. They can store it for some time, and when it is you can say more than a threshold level, it is excreted out into the spongocoel. So some scientists, they have this particular aspect of excretion with the help of the amoebocytes. The next part is the behavior of Sycon. So students, as we have discussed in paramecium, that paramecium had got lots of cilia over it. That's why it was a ciliate. So those cilia were moving all together in a similar fashion. So that was called as a synchronous rhythm, or a, you can say, metachronal rhythm. So all the cilia were moving together. But here in Sycon, all the flagellae of the choanocytes, they are moving independently. So there is no coordination. So there is no coordinatory behavior as shown in, as seen in the case of the Sycon. But the behavior, or you can say the coordination in between the body, see, the water is going out, the flagellae, they make a water current and they expel the water out of the osculum. So how this particular animal, it is knowing that water has to be expelled out of the osculum? So there is some kind of coordination which is contracting the whole of the body, which is contracting the cylinder, which is causing the contractions in the pinacoderm, and there is a specific water current formation. So there should be some kind of a nervous system which is present in Sycon. But scientists till now, they they have not found any kind of specific nervous system in case of Sycon. So they think that the cells which are present in the mesoglea, the cells which are known as coloncytes, these are responsible for the overall behavior of the Sycon. So this is the view of some of the scientists, that coloncytes, they may function as neurons in the case of Sycon. So if we discuss the structure of the coloncytes, so this is a type of the coloncyte. So we have discussed this particular cell in the case of the body walls, and we have discussed it as a star-shaped cell with some dendrite-like structures. So this is a typical coloncyte which is found inside the mesoglea of the Sycon, and this is, I think, very comparable to a neuron. And here what is absent is the axon part. So this is the cyton part of a neuron. It is very comparable to a cyton part. So some scientists, they think that these coloncytes, they are acting as the neurons in case of Sycon, and the movements or the behavior of the Sycon towards the external environment as a response to the external environment is due to these cells. If we take the example of another cells which are called as myocytes, so the myocytes, they are also showing contractions and relaxations. So myocytes were present on the ostia. So if the Sycon can judge that the myocytes they should be open to receive the water inside the incurrent canals, so there is some kind of nervous system. So this sense in the Sycon is formed due to the coloncytes. So this is another you can say a confirmatory kind of process. which confirms that the colonocytes, they act as neurons in case of Sycon. So students, that was all about the physiology of the Sycon, and these were all the systems that were going on in the Sycon. So in the next lectures we'll be taking up the reproduction process in the case of Sycon. So that will be the last part in this particular animal. So thank you for now.